Good evening and welcome to our second webinar of the year. Today we are very pleased to welcome Marie Baes, who is going to talk about pesticides that are more respectful of the environment and health. But first, I would like to take the opportunity to present ourselves and what our association is doing. We are the Association of Young Chemists of the Royal Chemical Society in the Federation Wallonia, Brussels. And our goal is to help young chemists, such as master's students, PhD students, and postdocs during their first steps in a scientific career. How? Well, mainly by organizing a variety of different events throughout the year, such as conferences, where we give you the opportunity to present your research and exchange with peers, by organizing lectures, such as the webinar of today, or lectures where we inform more about the best way to valorize your results and your personal skills. We also inform about job opportunities. We do company visits so that you can familiarize with the local chemical industry, and we publish a lot of job offers on our social media accounts. Our goal is really to create a link between academia and the local chemical industry, so that you have it easier to find a job or to start collaborations with industry. We also provide networking opportunities. Usually once a year we organize a badminton tournament, but as you know, for the moment with Corona, that's not possible. As many of the other events, neither. So we were looking for new kinds of things that we could organize. That's when we came up with a competition for the best video clip. This is an opportunity for researchers to present their work in an original, attractive video of 3 to 5 minutes for a scientific audience. This competition is open to all chemistry PhD students and is worth credits. And the best video clip will be awarded a big prize. So if you're interested, there is still time to participate. The deadline is March the 1st. I know that's not a long time, but for the moment there are not a lot of participants, so your chance to gain and to win 200 euros is really big, so think about it. If you want more information, just send us an email or have a look on our social media accounts on Facebook or LinkedIn. Now let's have a look at our webinar of the day. We are very pleased to welcome Marie Baes. She did her chemistry studies at the University of Namur, and afterwards she spent several years teaching, first at the UCL and later in Namur and Fleurus. In 2012, she started a new position as researcher at the Walloon Center for Agronomic Research. The CRRW is a very big employer in Wallonia, engaging beneath others many chemists. So Marie Baes became a laboratory and QC manager at the CRRW. And over the years, her research focused on various topics in the field of public health, such as biocides and the development of insecticides for the treatment of malaria. Today, she is going to speak about pesticides that are more respectful of the environment and health. So, dear public, feel free to post your questions in the QA section during the talk. At the end, we are going to try to answer as many as possible. And if you see a question that you are particularly interested in, just like it to let us know. So Marie, thank you very much for accepting this invitation, and the floor is yours.
So thank you very much, Miriam. So good afternoon, and thank you for participating to this call. I'm going to start the conference with a presentation of the Walloon Agricultural Research Center, which we call in shorter CRAW. After that, I will give an overview of the registration process of plant protection products in Europe and in Belgium, and of the analysis performed for the chemical physical characterization of pesticides products. I will continue with the presentation of some research projects in which CRAW is involved. Finally, I will present you the subject on which I work mainly, that is biocides used in public health to fight against vector-borne disease, mainly malaria. The Walloon Agricultural Research Center was founded in 1872 and is a public body of the government of the Walloon region in Belgium. The CRA brings together scientific research, service and support functions to Walloon farmers, breeders, horticulturists, forestry producers, and operators in the agri-food and related sector. I'm sorry. Its research projects involve Belgium and international partners, including universities such as the University of Namur and many other institutions. Now, a few figures. It employs approximately 440 people, including 120 scientists. The CRAW is located on 3 sites, Jean Blou, meaning being the main site, Libramont and Mussy-la-Ville. It operates 300 hectares of experimental fields, meadow and orchards, as well as beef and dairy cattle, pigs and poultry farms. 73 hectares are used for organic agriculture. It has 23 laboratories, 15 collections of genetic resources, and seeds of cultivated plants, and offers 60 types of service to the agricultural, food processing, and other related sectors. The research at CRAW focuses in 4 main areas. Firstly, precision agriculture, and secondly, precision livestock farming, both to produce in a more sustainable way. Thirdly, the risk management to stabilize production outcomes while protecting the environment and consumer, and finally, a better understanding products to ensure the quality of the production process and the derived products. The CRAW is divided in 4 departments, as you see in the screen. And I belong to the Knowledge and Valorization of Agricultural Products department. This department includes 3 research units, and I work in the unit focusing on protection products and control products, and studying residues and metabolites of these products in the environment and foodstuffs. These products are mainly, but not exclusively, pesticide products. Before going further, I would like to have a few words on what a pesticide is. A pesticide is a product designed to fight against a pest, that means a harmful organism. Pesticides are divided into 2 categories: plant protection products that protect crops against harmful organisms or that combat weeds. They are used in agriculture, of course, but also in horticulture, in gardens, public or private gardens, or for the maintenance of railway tracks, for example. The second category is the biocide products. Biocide products include disinfectant, some household products, disinfectant or products against moisture, algae, protection products, protecting not crops, but for example, wood or building materials, products to fight against harmful organisms, such as rats or insects, mosquito, flies, and etc. Now let's focus on plant protection products, PPP. Before they can be sold on the market, the product must receive approval from the authorities. I'm only going to talk about the procedure in Europe. At the European level, marketing authorization have been harmonized in 1994. The principle is that a product authorized or rejected in one country can be authorized or will be rejected in another member state for similar use under comparable agricultural and comparable climatic conditions. In this spirit, and also to share the workload, the member states are grouped into 3 zones with comparable agricultural, comparable plant health and environmental conditions, including climatic conditions. Belgium is in the central zone in yellow on the screen. In Belgium, the authorization of plant protection products is a federal competence. The competent authority is the Federal Public Service, Public Health, Food Safety and Environment. The committee consists of permanent members working in public institutions. The committee can always call on the collaboration of other competent persons, for example, from any laboratory or institutes specializing on the subject. 3 members of CRW works in this committee, 1 being a permanent member. Note that Belgium must first submit to the decision of Europe, but may after apply additional restriction for application or use. I'm now going to explain a little bit about the content of a submission dossier and when CRW and a chemist can be involved in it. To give you an idea, one dossier in hard copy contains 40 to 50 large folders, as you see on the screen. The procedures follow guidelines enacted by FAO, WHO, and OECD, as the analysis must be carried out according to good laboratory practices to ensure transparency and traceability of all results in the dossier. The first part of the dossier consists to determine the active ingredient content. The active ingredient is the chemical molecule responsible of the pesticide effects, thus which kills the pest. The nature and content of other components of the finished product is also determined when relevant. Relevant means that can have a toxic effect for the user or environment. The content of chemicals is usually determined by gas or liquid chromatography. This is one of the main activities of the laboratory where I work. This is typically an activity for a chemist. Another main activity of the laboratory is to test the essential physical and chemical properties of pesticide products in order to ensure safe and effective use of the product on the field. Not only the concentration of molecule is enough to characterize a product, but also well-defined chemical physical properties. The stability of the product during the time or at different temperature conditions are tested. The product is, for example, put in an oven during a period of time to simulate an aging. Real-time aging is also studied. Specific tests are carried out to ensure that the application of the product is as safe as possible, such as dust content, surface tension to be sure that the product is not too volatile, etc. We perform almost all tests in our laboratory. Other tests are carried out to make sure that an appropriate amount of active ingredient is homogeneously dispersed in order to have an event application. On the picture, you see, for example, a decanting of product, so the product will be rejected. It's also important to have good quality products to ensure a proper application with the sprayer. For example, too large particles or insoluble particles can cause a blocking of the sprayer. Keep in mind that if a spraying does not occur as it should, this results in a major risk of environmental pollution. Here are two pictures of the test that we do at CRAW with a sieve or using laser diffraction. Some tests, such as flowability, pourability, or dustability, are required to ensure that the product will flow freely or will be poured easily in the tank sprayer. For example, the test of pourability shows that not too much of product remains in the container. This unusual part of the product is a source of environmental pollution. If a product fails for one test, it is rejected. There are many other tests used to check the quality of pesticide products, but I'm not going to mention them all. You are welcome in our lab if you want to see all of them. A very short but very important chapter. It defines how the product will be used in practice. Let's take, for example, a fungicide used at 1 liter per hectare on potato against mildew, 3 application maximum at minimum interval of 7 days for a pre-harvest interval of 14 days. This is the basis for all subsequent evaluation in toxicology, residues, environment, and ecotoxicology. Then, the product has to be correctly labeled. The file must contain all safety data, all precautionary measures for humans and animals, the emergency measures in case of an accident, a detailed procedure for the destruction or decontamination of the plant protection product and its packaging. The compatibility with each type of packaging used is also verified. We do it routinely in our laboratory. In order for a product to be accepted, it is necessary to provide all analytical methods to be able to measure the component in the product, but also to measure its residues and metabolites in the environment. These methods have to be validated to ensure their robustness and reliability. Development of these analytical methods is very close to research. It uses advanced technologies. After that, the firm must justify the nature and extent of the benefits provided by the product. New products are always compared with reference products already authorized for similar use. They must have a similar or superior efficiency to the reference. There is therefore an evolution towards ever more efficient products. All negative effects on the quality of the crop are also reviewed. Note that the CRAW is officially recognized by authorities to conduct these tests. A very important step in the study of toxicology and metabolism of the product for user, animal, and environment at short and long term. To illustrate this point, I will present you two projects in which CRAW is involved. The Expo project aims to assess the average exposure of the Walloon population to pesticide in the atmosphere. The CRAW has developed a device shown on the slide to collect and measure the concentrations of 46 different pesticides in the air from around 12 sampling stations over the course of 1 year. The Propel PPP study's objective is to measure the drift of plant protection products near agricultural plots due to the wind, to the material, etc., in order to determine the risk of inhalation and cutaneous exposure by neighboring residents. These projects want to check the effectiveness of recommendations aiming to reduce the risk of local residents' exposure, such as the minimum distance between the field and the resident, exposure of a hedge, or the time for spraying during the day. Now, the assessment of residues and metabolites. The first step is to understand how the substance transforms and distributes itself in different parts of the plant, especially the edible parts. The determination of maximum residue limits is an important step. It is used to determine the concentration of residues likely to be found in foodstuffs as a result of the application. There are typically 8 trial fields for crops in Northern Europe to take into account a variety of weather and environmental situations. It is then necessary to estimate the acceptability of the residues. It means to check that a standard consumer eating the food at the threshold of the maximum residue limits is not exposed to any risk. Residues and metabolites are measured in food and feed. Baes is a vast project in which Dossier W is involved that aims to study pesticide residues in pollen and bee bread. Now, the behavior in environment. A product may be marketed for 10 to 20 years and will therefore be dispersed during that time in the environment, especially in soil and water. We study the rate of degradation in the environment. A product that does not degrade quickly enough will be rejected. The objective of Biodien project is firstly to determine the endocrine disruptor content in the water cycle in Wallonia and the Brussels region. Secondly, the project aims to create a risk anticipation management tool. The aim of the project Solfili is to evaluate the future of plant protection products in relation to cultural practices, mainly for a better management of underground water resources and their contamination. The goal is to develop an eco-responsible agriculture. Now, a short digression into this presentation to illustrate the performance of pesticide residues analysis method. Imagine a sugar cube dissolved in 10 Olympic swimming pool. We will find it. This corresponds to 5 grams of substance dissolved in more than 30 million liters of water. Keep this in mind when you hear that substance are being found in the environment. Finally, the ecotoxicology is assessed. The ecotoxicology is the evaluation of the effects of the active substance on target organisms. These organisms are chosen because they are representative and ecologically important. You have birds, mammals, pollinating insects such as bees, aquatic organisms, arthropods, etc. A risk assessment is evaluated for each type of organism. I stop here for the PPPs, their characteristic and research project. I'm going to focus now on totally different types of pesticides. This time, biocide products will represent my main activity. I'm going to talk you about control products to fight against malaria. Malaria is a disease caused by parasites that are transmitted to people through mosquitoes. That is why we call that a vector-borne disease. In the year 2019, there were an estimated 229 million cases of malaria worldwide. The estimated number of malaria deaths stood at 409,000, mainly in Africa. Children aged under 5 years are the most vulnerable group affected by malaria. They accounted for 67% of all malaria deaths. Nearly the half of the world's population is at risk of contracting malaria. Some population groups are at considerably higher risk than others or can develop severe disease. These groups include children under 5 years of age, pregnant women, and patients with AIDS. Vector control is the main way to prevent and reduce malaria transmission. In other words, this aims to kill mosquitoes. Two forms of vector control, insecticide-treated mosquito nets and indoor residual spraying, are effective in a wide range of circumstances. Indoor residual spraying consists of the application of an insecticide on the walls of houses and huts. These insecticides need, of course, to be good quality products. The quality control of these products includes to check physical chemical properties of the product that are very similar to the property already seen in the previous part of the talk. I'm not going to talk about it again. Criteria and limits are established by the World Health Organization. The second way to prevent malaria is the use of long-lasting insecticidal nets. They are insecticide-treated nets, usually bed nets, that provides a physical and chemical barrier to insects. The release of the insecticide is controlled, not too slowly and not too quickly. Now, I'm going to explain how mosquito nets are long-lasting. Nets used to fight against malaria are not just nets that are dipped into a liquid containing an insecticide. Other nets have been developed which are more efficient, safer for user, and safer for environment. The nets and thus long-lasting nets means that they are designed to remain efficient after at least 20 washes and at least 3 years of recommended use. Recommended means not to use it as, for example, fishing fillets. There are two main types of long-lasting mosquito nets, coated and incorporated nets. For coated nets, the insecticide is in the surface of the yarn and is coated with a binder. With an incorporated net, the insecticide was incorporated into the yarn before being woven. This insecticide can migrate gradually to the surface. As this is a specific product, a textile plus an insecticide, it provides both mechanical and chemical barrier to the mosquitoes. These products have their specific test: netting mesh size, where we count the number of holes per square meter, dimensional stability to washing to make sure that the net will not shrink too much or deform after washing. We test also the burning strength to make sure that the net and the seams are robust enough, and also the flammability. Regarding the chemical properties, we determine the active ingredient content by gas or liquid chromatography and how much insecticide remains after several washes. This is for the wash resistance index of the active ingredient. To test the release rate of the insecticide, we have to wash the net. The washing is fully standardized. That is why we prepare ourselves the soap and do it by hand and not by putting the net in a washing machine. How to do that? A piece of net is put in a glass bottle with soap, washed by inverting the bottle by hand, then rinsed and dried during 22 hours. During this time, the active ingredient can migrate to the surface of the net. It has to be done 4 times for quality control, sometimes up to 25 times. And after, the pieces are cut with scissors and the insecticide content is determined. This is to evaluate the content of insecticide remaining after several washes. At CRAW, we perform many quality controls and analysis of mosquito nets and products used for indoor residual spraying. We are one of the world's reference laboratory for quality control of pesticide products used against vector-borne diseases. I will now conclude with one optimistic thing, is that thanks to prevention treatment, anti-malaria program, diagnostic tools, survey, and all the others, the number of deaths due to malaria has been reduced by half in 15 years. Thank you very much for your attention. And now I'm waiting for your question.
Thank you, Marie, for your presentation. There are some questions from the audience. The first question is Dossier under ISO regulation, and if yes, what kind of regulation?
So we have good laboratory practice certified for registration of products. So for plant protection products that will be put on the market. For the quality control and analysis of mosquito nets, we are ISO the ciphers, the good ciphers is ISO 17025 ISO accredited. In fact, it is the accreditation for laboratories performing analysis on a routine basis for quality control, for example, chemical analysis, I mean.
Okay, thank you for your answer. The second question is, are the long-lasting nets expensive, as they are mostly used in the poor countries? So,
usually, in fact, you have a lot of help provided by the United Nations. There are a lot of programs to provide mosquito nets for free to local population, but usually a net costs $1 or $2, so it's not so expensive.
Okay, perfect for these poor countries. So the next question is firstly the people. Thank you for the presentation. It was for him an interesting presentation, and for all of us a very clear presentation. The question is, as at the beginning you mentioned that one of the acts of your research was valorization of agricultural pesticide. What did you mean by that? Are you looking for other applications without the product transformation, or are you considering chemical transformation of the pesticide currently under European regulatory treat to make them suitable for more suitable application?
So no, we are not developing pesticide product. In fact, we only analyze them. So I don't know if this answers to your question, but we are also testing new products such as biostimulants and other products that are used in organic agriculture. So all these products are innovative and try to reduce the impact of environment on during the application and after. So I don't know if this answers to your question.
I think yes. The next question is about the lasting net. Is the physical barrier not enough to stop the mosquito? Is there an added value to use a pesticide on it? Is the mosquito dead or dying when it is in the contact with the pesticide?
So yes, the mechanical barrier is useful only if you are in the bed. So if you I don't know for what, if you get up out of your bed to go outside, I don't know for what, so you can be bitten by a mosquito. And in fact, the principle, the idea behind that, the use of an insecticide on the net, is to reduce the population of mosquitoes. Imagine that in our countries, during the summer, we have one mosquito in our bedroom. In African country, it is 1,000 mosquito in the bedroom. So that is why we try to reduce the population of mosquitoes by using an insecticide. Okay, and yes, a mosquito is well killed when when the mosquito is in contact with a mosquito net. Yeah.
Thank you for your complete answer. We have a lot of questions. So the next question is how the PPPs tested on pollinators.
On what? I'm sorry, I didn't.
Or are the PPP tested on the pollinators?
On the poly what? I'm not sure that I understood.
The pollinators. I think the.
Oh yes, on bees, for example.
Yeah, yes. Yeah, for example.
So we have in CRW. I'm sorry, I'm not involved in that project, so I will try to answer as I can. So I know that in CRW we have a huge project to study bees. So we have some partners who have these bees that are in contact with several cultures, and we test the amount of pesticides in the pollen, in the bee bread, and also in the bees. So we kill the bees, sorry, and we test it.
Okay.
Yes, and we have several types of bees. So bees in contact with agriculture, the bees in the center of a forest, for example, bees in the city. So we have a comparison of all this kind of population to compare the effect of agriculture on bees.
Okay, thank you for your answer. And the last one is the net technology and testing also available against tsetse fly? I don't know if you have an answer for that.
For tsetse fly, I'm sorry, I don't know. I don't think that the mosquito nests are tested on tsetse fly. In fact, this is totally not the aim of a mosquito net to kill other insects than mosquitoes. So I'm not sure that it is efficient against tsetse flies. No. because why mosquito net, a bed net is used? Because the mosquito that transmit malaria bites during the night. That is why we used mostly bed nets. But for TT fly, I imagine, I'm not sure, I imagine that you are bitten during the day, so I'm not sure that mosquito nets are efficient.
Okay, thank you for all your answer, and thank you for this presentation, really clear and complete. I leave the micro to Miriam to conclude. I don't know.
Thank you very much, Manon. Thank you very much, Marie Baes. It was really, really a nice talk. Thanks for being with us. And I have nothing else to say than to wish everyone a very nice evening and to see you next time. Thanks a lot.
Thanks a lot too.